Good morning to me because I'm in California. It's in the West Coast. But good afternoon and good day to the rest of the folks and everyone tuning in from around the world. Um, Thank you for joining us, and today for celebrating the Day of Immunology. And our panel is really going to talk about immune responses to vaccines and pathogens across age groups. And so I'm going to be moderating the discussion today, and just a little bit of background. So my name is Colwyn Headley, or you can call me Dr. Coco. And I am a postdoctoral scientist at Stanford. I am studying the impact of aging on mitochondrial dysfunction within the immune cardiovascular axis. I got my PhD from the Ohio State University, and some of my work has focused on the impact of aging on host-pathogen interactions during infections. And some of the pathogens included Mycobacterium tuberculosis, Mycobacterium avium, influenza, and a tinge of SARS-CoV-2 or COVID-19. And T cells are my favorite immunes. And so we've got an incredible panel today for you. And that was just kind of break down the nuances introduced by natural biological aging into immune response against infections. And so I'll introduce panelists first, and then we can just jump into questions. Sounds good? So first we have Dr. Fabien Prodjinotho. Dr. Prodjinotho is a senior scientist in translational immunology and immunotherapy at the Technical University of Munich. His work focuses on integrating immunology, parasitology, neglected tropical diseases to kind of explore how immune regulation and look for and impacts of therapeutic implications. So he's originally from Benin, but I'm not butchering that. Earned his master's in immunology and physiology, and he did his PhD at the University Clinic Bonn, studying antibody suppression in helminths. He's deeply committed to capacity building in immunology research between North and South, and within South itself. He's a co-founder of the Beninese Immunological Society, and which is geared at promoting sustainable research networks in sub-Saharan Africa. We also have Dr. Oyebola Oyesola. Dr. Oyesola is currently an independent research scholar at NIH, the National Institute of Health here in the USA, and her group, the Immune Planning Unit, is focused on understanding how previous infections and exposure to environmental antigens can reprogram the immune system and influence outcomes to subsequent injury. Dr. Oyesola was previously a visiting scholar at NIH, where her research focused on individual macro and micronutrients and genetic factors that sort of influence susceptibility to infections. She earned her PhD from Cornell in immunology, and she also has a master's and a DEA. You got all the degrees, which is why we're here. You got to put respect on our names. So we also have Dr. Oladipo, Kolawole Oladipo. He's an international fellow at UCMR. He got his PhD in medical virology immunology from La Doke Akintola University in Technology. He holds a faculty position at the Adeleke University in Nigeria. He focuses on vaccines and diagnostic kits, diagnostic kit development for tropical diseases, for neglected tropical diseases. We also have Dr. Adam Abdullahi, who's currently a Takami scholar in international health at Harvard T-Chan School of Public Health. He's also a research fellow at the University of Cambridge on both coasts. He has a background in tropical diseases and infections, infectious diseases. He's currently researching, or his focus is looking at understanding population level determinants of HIV treatment outcomes during the impact of viral immunosuppression, emerging drug resistance, and core viral infections. He's also interested in evaluating vaccine responses to emerging pathogens across African populations where vaccine access is kind of limited and sparse, with the aim of generating quickly empirical evidence to guide infection policy. And so we very high level, but we're just going to begin just kind of breaking down for the folks that might tune in eventually or that are in here that are not completely familiar with the immune system or what a vaccine is or why we should even care about aging, even though these are, we're all getting older. What is the immune system? Who wants to tackle that one first?
I can get started. I think just simply said, I would say the immune system are like the soldiers of the body, right? It's like the body's defense system, and they help protect the body against foreign agents, and this could be antigens and not necessarily infectious agents. So they're the body defense system, but you can also have them also be involved in when you have an exuberant immune response, being involved in autoimmune disorders as well. So they have to be a balance, I guess. So they're mostly defense, but you have the other side as well, where they can sometimes be harmful, either in allergies or autoimmune disorders.
Adam?
So I'm happy to just add something to that. So yes, the immune system is really a defense system, but just to note, it's a really, really old system. It's archaic. So traditionally speaking, there are certain things that your immune system, which is this defense system, would come across, would be able to sort of take care of by clearing. But there are also certain pathogens that it would recognize that it hasn't sort of seen this before, and it has to go through sort of a different kind of process to be able to put forth these defenses up. So it's archaic, it's really old. It wouldn't necessarily respond to everything. It would be very selective in terms of what it responds to.
Yeah, and I guess we can segue into response, right? So why are vaccines such an important thing for human health?
Yeah, so maybe I can add on that. So why vaccine are quite important. So as my colleagues and the panelists mentioned so far, so our immune system defend ourselves, right, against foreign antigen. And then we are talking about foreign antigen here, antigen, so which is what the immune system recognizes to drive any immune response. Now to really help the immune system to go that way, because our immune system does not actually be in contact of all kind of antigen. So we need to prepare the immune system to recognize quickly and then to react in an efficient way, right? So, and then vaccines are there to prevent, at least to build our immune system in that way, that we give antigen to the immune system to recognize and then to develop memory, so that when we have this antigen in real life, the immune system can quickly react to that. So put it in a really simple way, this is how vaccine help human and also the immune system to quickly then defend our body. Yeah.
I guess
Yeah, I think
Dr. Oladipo?
okay. I think to add to what my brother he said, some of the time we have what we call natural acquired immunity and artificial acquired immunity. So what we do, it naturally have been used to this pathogen naturally, that's maybe exposure through any form, various means, and I've developed a system whereby when I recognize such pathogen or antigen in the future, I should be able to fight. I used to use this analogy to my students that, okay, I'm a soldier, I have a gun, I know this is where my enemy is coming from, and this is kind of a bullet I can use to kill the enemy so that the enemy will not harm me. So now what we now do in vaccine is that we create an artificial system that in case if your enemy is coming to this side that you have never seen before, how did you attack the enemy? So the vaccine is a way of training our immune systems in recognizing unseen enemy before, that when such an enemy we come in contact with you, you should be able to knock it down, destroy the enemy. And that's just the simplest way I can just explain for.
That's a great analogy. Dr. Oyesola, you wanted to add a point?
Yeah, exactly the same analogy about soldiers, and so I would just not flog that again. I think Dr. Kolawole did a good job.
So on the topic of soldiers, so I mean, if you join the army, you got to go through basic training, right? So there are certain things that make you a good soldier. So in sort of expanding on this analogy, then what are some things that make a good vaccine a good vaccine? Like what are some qualities that help protect? What qualities does a vaccine instill that makes it protective against pathogens?
Yeah, I think I'll just say first point where I will allow my panelists to take first. The first thing criteria are, because I venture to vaccine development, it's the safety. It must be safe, because there's tendency for your vaccine to be highly immunogenic but not safe. So one of the number one criteria of a good vaccine is to be safe for all ages. And that's one of the best, that for the kids, the pediatrics, for the youth, for the elderly one, for the immunocompromised, you can say that that vaccine is safe for everyone to be used. And the second point I will raise is ability to elicit a great immune response, because the degree of immune response that your vaccines are stimulate or produce has a long way to go. Categorically, the IgG and IgA, so that it will have a long-lasting effect on the body system. You see, for instance, in Africa, there was a time you're using yellow fever vaccine that after 10 years, you need to get another shot. But now, currently now, the vaccine we have for yellow fever, once you get a shot, you don't need to get any booster again. It covers you for the end of the entirety of your life. You can see that this makes this vaccine a better one.
Yes, we can dial back on that really quickly. I like to think about it from the aspect that there's something they call the innate immune system, which is kind of like the first response that the body, and then you have the adaptive immune response, which is really, so there's the question about memory, which we kind of got into when we were talking about vaccines. So a vaccine should be able to induce a memory response, right? Because it's the training and you want to be able to induce an effective response. So typically, I guess people try to focus on the adaptive immune response, so generation of T and B cell immune responses, which are the key signs of an adaptive immune response. But I think there's now some interest in innate memory, but I think generally a good adaptive immune response, B and T cell response, antibody responses are critical for an effective vaccine response.
Yeah, so just to touch up upon, two interesting points that, the previous panelists have spoken about. So the first thing really is safety. And that means that you really want whatever vaccine you go on to develop not to sort of elicit some sort of adverse immune responses, which it's really quite difficult to come across, even across a wide range of sort of currently licensed vaccines. Sometimes you get people still having anaphylactic sort of responses, so to speak. So that's one, safety. And the second point that they really talk about is the immunogenicity, and that's really has your vaccine will be able to elicit two components. So that's your innate immune response, but also sort of the adaptive immune response. And the adaptive immune response, there's the B and the T cell component to it, really. So B cells, you really care about antibody responses, which often you can really measure. In that way, you can look at sort of neutralizing antibody responses. But also in recent time, we found that the antibody don't necessarily directly protect. There's also another component called sort of antibody-dependent cellular cytotoxicity, so to speak, or ADCP or ADCC. And these also kind of act to contribute to sort of that response. And then, of course, there's the component, which is the cellular component. And then you're looking at, your T-cell responses. You want your vaccine to really have a very good balance of pro-inflammation. So what's happening in terms of, putting under control, but also anti-inflammation, just to make sure you don't have too much of either. And probably the final point that I would point out in terms of what a very good vaccine should compromise of would be efficacy. So in this case, and you measure this really differently. So for example, for the SARS-CoV-2 pandemic, when that happened, we're talking about the efficacy of a vaccine would be, for example, if the outcome is to ensure people don't have severe outcomes, it means that sort of population are protected against the severe outcome, whether if it's hospitalization or death. And I'm bringing this because I did a lot of work on SARS-CoV-2, looking at immune responses. And I had, this elderly population in Cambridge in the UK that were all above the age of 70. We found that after the first dose, they didn't do well. After the second dose, they did well, sort of still lower than people who were around the age of sort of 30. But we gave them a third dose of the vaccines, which kind of equilibrated the responses. But essentially, all of them got re-exposed to SARS-CoV-2, but they never had sort of a severe outcome. None of them ever had to come back to the hospital, which under these circumstances, you would say that the vaccines, especially with the boosters, were relatively efficacious. The neutralizing responses were there, but it didn't necessarily prevent the infection from happening again. So it's really multifactorial, multidynamic. Just thought to add that in there.
So maybe I'm gonna add on that. Of course, so this is specific a general aspect of a vaccine, a good vaccine and vaccine response. But then when you dive in and then you come to specificity and so on. For me, my conception nowadays of a good vaccine is a vaccine that, of course, then display those characteristics, but also is adapted to a given population of age group and also across a region, because you cannot have, nowadays we're going to dive into that later on, but you cannot have an ideal vaccine which works across all the regions and all the population. Is impossible, because we differently react and so on and so on. So for me, a good vaccine is a vaccine which is really adapted to a population exposed to a given environment, and also being in a certain range of age that we really master and already identify how the immune response reacts, because you always adapt your vaccine to the immune response. So you have general conception of vaccine and vaccine response, but a good vaccine for me should be really adapted to a given population and with characteristics that we need to define up front.
That's a great point. And so just to recap, and I'm gonna jump back to the army analogy because I really like it. So if you imagine that the immune system is really this army, for just breaking down the concepts of immunology, or like what the immune system does, you've got sort of like the early responders, right? Sort of the infantry, which is like the innate immune response, right? The first responders to a pathogen in your body. And then you've got really the snipers, or the adaptive immune response, which is like super specific, right? And that's kind of what we're alluding for. And so the infantry is those are the folks that spot the enemy at first, and they're like, hey, yo, we need backup, and they'll communicate to the snipers. The snipers now they're very specific, and they'll, if trained properly, right, which is what a vaccine is supposed to do, it's going to be able to take out the target. But that doesn't always happen, right? And as Dr. Prodjinotho was alluding to, like with different population ages, as well as Abdullah, like with different ages, there are different efficacies of vaccine. And so that's a good segue into talking about like the aging process, right? And so for me, I see aging, and I'll use another analogy because I think analogies are the easiest way to really like get conversations going. But I kind of look at aging from the perspective, immune aging, particularly in the perspective like a vehicle, right? So with a young immune system, like really a brand new car, where it can really weather the elements. You can drive, it's got good brakes, good gas mileage, right? And then as you get older, right, your immune system kind of resembles this clunker where the brakes don't really work that well. The engine, I don't know. You don't want to get caught with it on a highway. So in segueing from defining what a vaccine does, then maybe we can talk about what are the sort of the nuances that changes as the nuances that changes with the immune system as we get older, and how that influences like vaccine efficacy.
I can start on that. I think it all, like you were alluding to, it's like a brand new car, or you'll say some tabula rasa. So you've not seen anything before. And so your immune system is basically very naive, very immature. So I actually think about it like a bell curve, because you have two people at the extreme ends. You have the neonates, and then you have the old, and then you have the young adult or young sort of population. At the very extreme end, you have like the neonates is very immature. So at that point, you probably also need to boost the immune system very well, to train it very well to be able to recognize antigen, because, all those T cells have not seen anything before. Mostly it's just coming from the mother, and then at the far end you also you've seen too many things, and so you're kind of overpowered, and so everybody's distracted. There's so much distraction going on. Meanwhile, at the middle point you have sort of like, yeah, I'm good, I can see things, I can be trained, and I can respond effectively. So I think about it like a bell curve. And what are the factors that kind of influence this variation? Like Dr. Fabien was kind of alluding to, you have the environment. So where you're living, where you're staying, can dictate the kind of antigens you've been exposed to in your lifetime. And so all of these factors, environment, are some of the factors that can change how the different age group in different society or different regions can respond to a vaccine, which is why it's important to kind of discuss efficacy or effectiveness of a vaccine from a global standpoint rather than from a regional standpoint, I guess. I'll be interested in continuing that conversation, but I think it's important. I would like to give other panelists also the opportunity to share their thoughts.
Well, I think I just have a little bit to contribute here. One of the factors, like Dr. Oyebola rightly said, is that I'll be talking from the aspect of vaccine development. Some of the time, the pathogen of development, there is something we should evaluate population coverage at the point at which your vaccine is being developed. You understand that the pathogen, the region where the pathogen is obtained from in the vaccine development has an impact in the efficacy according to region, or according to race, or according to gender, because all these has a role to play. That's why I support what my brother said, Fabien, that it's multifactorial. There was a time rotavirus vaccine was introduced in my country, Nigeria, Africa generally. That vaccine wasn't performing well like the way it was performing in Europe. So we discovered then that from literature that the isolates used then were mostly obtained from that region. So for the new one now, they have to incorporate other pathogens from other African region in order to increase the efficacy of this vaccine. And like Riley said, Dr. Oyebola Riley said, what we have been exposed to at the early stage of our life also matter. I can cite the example of SARS-CoV-2, that some of the time in African population, we have been exposed to some pathogen unknowingly, and we have developed a level of immunity against it. It's only that I couldn't move further because of funding. During the SARS-CoV-2, I was trying to measure the response of antibodies to individuals that have been vaccinated, who took first shot, second and third, and naive individuals that have not been vaccinated. And you discover that even those who are not vaccinated, they have enough neutralizing antibodies at the same part of those who have been vaccinated. The only challenge we have is that we don't know how long will it last in the system for them to. You can see that for that region, what we are having is quite different. And when we talk about vaccine efficacy, it's regional based. It is a multifactorial factor that a lot of rules, a lot of factors will come into play. And that's just my own thoughts about it.
So before the other panels, so let me see if I can maybe break it down just a little bit. So you're saying that in context of vaccine development, there's and this is from more so from like a not just like the basic or translational research perspective, but actually like industrial level vaccine development, right? There are issues or there are nuances with figuring out what is the right antigen or right what's the it's not subtype the right variant,
the antigen,
right?
epitope of the antigens to be included that can stimulate immune response.
Right, and so how before we even get to that point, for there's like a tinge of I hate confusing folks. So in development of antigen response, right? So like in development of your body responding to a vaccine, typically you have your innate immune response. It will first detect or capture the antigen or the pathogen or whatever, and then it will fragment it, right? Process it, and then present it as fragments to the adaptive immune cells, which is like your T cells or your B cells, etc. And so the nuances come into play here because depending on the variant or the subtype of the pathogen or whatever, there's differences in the expression patterns of these fragments, right? And that's what contributes to the variances in immune response. That's one part of it, right? And the other part is just human heterogeneity. Is that
exactly?
Good.
So maybe I'm gonna add on that about immune response over aging and so on, and how, I mean, you just mentioned that your APCs, antigen presenting cells, should then recognize and then process the antigen to present to the other team, mostly T cell, then to feed the B cell to produce antibody and so on. But then so that those antigen presenting cells recognize, they should be capacitated, right? And then as Dr. Oladipo mentioned, there are some of those antigen or parasites which render those antigen presenting cells non-capable. So they are more of this tolerogenic phenotype. They tolerate the presence of the antigen. Then they cannot really present the antigen in an efficient way. Whenever they cannot present the antigen in an efficient way, you get what we called, instead of getting effector cells to power the immune system to clear the antigen and so on, you get a more regulatory T cells, because you were not able, the antigen presenting cells were not able to efficiently present the antigen to activate the effector T cells. But the most important thing with aging, just to come back on that, is that when you get old, your immune reactivity decline. Why? Because you have what we call immunosenescence. So the cell also get old and then they get exhausted. So they cannot really work properly. And in old people, in addition to this natural immunosenescence, so they also produce, so there is an involution of the, maybe I'm using too much of those immunology term, but then there is an involution of what we call primary lymphoid organ. So which means that they get involved, and whenever they get involved, so the tissue or the tissue which are supposed to play their role to produce cytokine or mediator, and so they dysfunction, and then you have a lot of inflammatory mediators which are produced, because even there is a degradation of the tissue which release self-antigen, and then you have so many inflammation ongoing, what people called inflammaging and so on and so on. So which means in elder people, so they they have less ability in the T cell or whatever than to power immune response or to react efficiently because of the cell get all immunosenescence, but at the same time, organ tissue involved with producing a lot of self-antigen, which somehow release inflammation and so on. This is something that we really find observing in COVID, where you have this initial storm in cytokine in elder people compared to younger people, right? So is just to add on that, why this is happening in older people, and then how the immune system function then involved in decline over time when it comes to aging.
Yeah. So just to add something really there, these sort of phenomenon that he's describing actually happens on either side of the spectrum. So one particular pathogen of interest would be your hepatitis B virus. And so if you're under, for example, the age of 5, 6, 7, and you get exposed to hepatitis B, which I've done a lot of work on hepatitis B sort of in West Africa, is inadvertent. So young children just play around and they get exposed. And for some reason, their immune system is so immature that it can't really detect that it's a pathogen. So it doesn't really respond to it at all. So you become chronically infected with hepatitis B throughout the duration of your life, just for the simple fact that you got exposed when you were less than, for example, age 7. For those people who get exposed after age 10, within 6 months, they automatically clear the virus. And yes, their blood markers would show that they've been exposed, but they don't become chronic carriers. So it happens on either side. And I think that's really the interesting thing about the immune system, is that pediatrics, until a certain age, it's absolutely immature. It doesn't have sufficient enough sort of B cells, T cells. Affinity maturation can't happen. Somatic hypermutation is not going to happen. Sort of the antibody shiftings would really not happen. So under certain circumstances, it can't respond well. And also on the other side, immune senescence happens because essentially it's been exposed to so much that at that time it's just too tired. And we've seen that with, SARS-CoV-2 again, like in certain populations, they had to have 3, 4 vaccine doses before it became 1 or 2 doses for people who are, within the age range of what you would say healthy adults and have a robust immune system. So just to point that out, and I think that's really interesting overall when you talk about the immune system, especially in the context of aging.
Yeah, and I guess I'm gonna bring it back to the army analogy. So we can sort of look at the sort of how aging contributes to sort of the friendly fire within the body, right? Like that's really what happens when the vaccine's supposed to help protect you, but the soldiers end up shooting at themselves, kind of in a sense. And aging, right? The natural process of biological aging contributes to that, just because things just stop working properly. But also, it can be argued that there are other factors, like we mentioned earlier, the environment, and maybe comorbidities that contribute to a decline in vaccine efficacy. So do you guys want to comment on that? Comment on, first, what are comorbidities, right? Do people understand that?
Yeah, so happy to take that.
Yeah. Go ahead, please. Yeah.
Right. So comorbidities would in this context would be so there are people who would be typically sort of healthy and have no underlying conditions. So for example, as you age, you may have diabetes, hypertension, or you may be a bit significantly younger, but you have kidney disease, sort of liver disease, and these significantly do contribute to how people would respond to vaccines. So these are probably one of the most significant factors that determines how well people would respond to vaccines or not.
Yeah, I think, like you rightly said, I would talk from the point of a microbiologist now, that when we talk about comorbidity is when you have 2 major infectious disease co-infecting a single individual. For instance, someone that is already infected with HIV and later discover that he or she is infected with hepatitis B or has HPV, which is human papilloma virus. We can see that so I'll go back to my analogy. like you have one enemy to attack. Now you now have two enemies to attack. You can see that you have more war that the system, the entire city is the body is fighting. It's different from when I have a single enemy that I'm fighting. You know, this enemy is coming with let me say a pistol, and this one is coming with another brand of gun, like let's say AK-47. So it means that the way I will respond to the two enemies now is kind of different, because they are jointly attacking me, and my responses will kind of different from someone that is only having one, and that is a form of comorbidities. And some of the time we have seen cases in patients that have more than two disease conditions that somebody is fighting with. So their responses to vaccine will be quite different.
Yeah, just quickly, I know that we're short of time, but really quickly saying that I agree with both parties that when you say morbid, it's sickness, right? It can be an infectious agent, or it can be something chronic, right? So it could also be, as Dr. Adam kind of alluded to, like it could be being obese as well. It could be diabetic. So something else you're dealing with, either infectious or non-infectious. And that can influence the trajectory of another subsequent injury, right? And that can be, depending on what it is, can be bad or sometimes even helpful. So yeah, just starting.
Okay, I guess we'll move on to the next question. So, I mean, I know that comorbidities, some of the major ones are related to the cardiovascular system, and cardiovascular disease in itself are probably #1 killer. And we know that these dampen or pretty much like reduce your ability to your immune system to respond. And so what are some strategies, some pragmatic things that individual can do to help boost or current research that's going on to help influence immune response, to improve vaccine efficacy, especially for the elderly that may have comorbidities?
Well, maybe I can already start here. I mean, the immune response to vaccine always depends on the immune cells, right? So recognition of the antigen and so on. So in immunocompetent individuals. So, and then this is also, I mean, you have two arms that you have to take in account here that, I mean, we already mentioned environment and also co-infection, which might really completely decline the response to the vaccine. So there have been some systematic review, really nice systematic review showing that as closer you come to the equator, or I mean tropical regions, so the vaccine efficacy completely dropped down. And then those regions are always exposed to helminth infection or exposed to other pathogens and so on and so on. And so which means that environment and exposure really play an important role. But now when we come to the older people, that immunosenescence then does not help, or I mean exhaustion of the immune cells. So they work, as Dr. Adam said, so they work a lot and then they are really tired. They cannot respond anymore. And then how can we do now to fit them back so that they can really work? I mean, you have non-specific measure like good practice in health, like nutrition, and also good sleeping, sport, and so on. But then you have also research proven method that really show how this can work. I mean, there has been a study trial in US, I think so, about a natural compound from a bacteria, I think, or fungi and so on, rapamycin, and then they really treat people with rapamycin, and then it show clearly that then after, when you challenge the whole immune system with influenza, their the old person receiving rapamycin beforehand, the immune system is really fit compared to the placebo. So which means that you can go away using even natural compound to do this. Or what people selectively doing today is to capacitate the vaccine with what we call adjuvant. So you add some molecule to the vaccine so that the immune system can strongly or can recognize better, and then you have more deposition of the antibody into the peripheral system to respond better. And then this gonna last longer because you have really huge deposition of the antigen that those adjuvant help to keep then going. And then the immune system, even in elder people, can be reacting really efficiently and then quickly. You can also do some what we call adoptive transfer of immune cells or blood transfusion and so on and so on. There are many things ongoing about that, but what I want to say at the end is you have non-research based practice for sport or nutrition, because the nutrition, what does the nutrition do is just to activate the metabolic part of the immune system. And all our immune system rely on metabolic function, right? Whenever you do not have metabolism going in a cell, the cell won't be capacity, won't be active, right? And then in addition to that, you have what I just mentioned now about all those research-based technology like adjuvant, vaccine, and so on and so on. So that's what I can mention here.
Okay, I think I want to talk about non-research based factors that's so simple that can help from one. Riley said it made mention about nutrition. I'll be talking about sufficient sleep. If you sleep very well, it's going to enhance the immune response, our immunity. That comes with stress management. That's because when you undergo too much stress, there is a way it has impact on our immunity. So we should learn how to manage our stress so that we don't overstress ourselves, so that our cells are able to function well. And we have what we call there's a group of people that practice lifestyle medicine, like consumption of alcohol, some other things that are not good. All those things has a negative impact on the way we respond, our immune cells respond to things. And I used to smile. I remember properly that during COVID, we were talking about wash hands, hand sanitizer, all the stuff. The reason why I'm bringing it, I'm talking about proper hygiene. These are things. Now COVID has gone. That habit has reduced.
People forgot to wash their hands.
Yeah. You go to many places, marketplaces in my country, you can see where you can wash your hand, you can use a hand sanitizer. Now that nobody is talking about it, but all these things has a significant role to play because it reduces the rate at which you get in contact with the infectious pathogen that can be detrimental to our immune system. So these are points that I can raise that doesn't need sophisticated equipment to enhance our immunity so that we can respond well to vaccines.
And just before the other panelists, so because I like that you mentioned sleep and stress mitigation, how much sleep is a good amount for the lay audience?
Well, I would love my other panelists to talk about that. We are all experts in that field. You know, naturally we see a lot of things. I do tell people that they will say that minimum of 8 hours you have to sleep daily, but when you have a lot of work in the lab, for myself, I don't get up to 8 hours sleep. But by the time you agree that you have lesser work to do, I think it's more advisable to have a minimum of 8 hours sleep per day, because that time you should have a system that is working in place. And you should be able not to stress yourself like the way we are doing. But I know it's a bad habit that I'm also working on to increase the number of hours I sleep, and having a stress management system in place to manage my stress, because there is no way we not do with stress, but you should have a good stress management skill to manage it. When we are overstretching ourselves, we should know the time to take rest, to go and sleep, and come back to the work.
Or just an outlet. So, for example, I go play soccer. I got a soccer game every Sunday. I go kick a ball around. Just every time I get out of labs, that's my like because I definitely do not get 8 hours of sleep. I'm maybe 5 or 6 right now. So I gotta cut about the information somehow. But yeah, so the other panelists, would you guys like to comment on some of the strategies to sort of boost immune response, and as Dr. Prodjinotho had mentioned, it really does metabolism. You are what you eat, so that definitely plays a huge role, and I do believe in that as well. That you have to have good power, and you have to charge the battery, which really what sleeping does. So
that's just a very quick comment from me. So yes, very well put. So exercise is really, really important. Physical activity, especially for people who have cardiovascular risk. Older population, typically the advice is get a whole lot of physical activities, move around, walk a lot. So just adding to that strategy. But there's in recent time, there's also been a lot of interest in sort of immunomodulatory therapies. And a few years ago, so this is obviously a more research sort of centered approach to how you can make vaccines better. And a few years ago now, the Nobel Prize was actually for characterizing, PD-L1 as an immune checkpoint, i.e., how can you manipulate the immune responses in certain way, either to sort of block them or to enhance them. So there's been a lot of research, of course, at the moment not available at population level for use, but there's certainly been extremely interesting sort of data to indicate that you can actually stimulate certain components of the immune system to allow it to sort of enhance response. T cells particularly, because there's evidence, especially sort of from HIV research, is that you can have, for example, neutralizing antibodies present, but if you don't have the T cell component, then you can't really sort of box a pathogen to be very well to be very well controlled. So probably that's just one of the more research-centered approaches to improving. So immunotherapies are, I think, an evolving sort of research area in terms of how to improve how vaccines and therapies in general control pathogens better.
Yeah, and I'll just add on to the research part. That I know that this conversation is really for a global audience and a very lay audience. So I just want to put a caution that no, I was not advising anyone to take advance or take a room for right. So these are research based. You have to make sure that you talk to your doctors and the public health professionals. So do not take information to enhance. But I think on a research level, of course, there are now research looking at adjuvant and how that can improve vaccine responses across board. And I think this kind of speaks more to the idea that research has to be global. You can't do research in just one region and assume that the vaccine responses and the clinical trials done in those regions will be enough for global use. So different populations in different places across the world needs to be. And then I feel like we'll also need to know what is normal in a region, right? What is normal in Nigeria? What is normal in Brazil? What is normal in Indonesia? And how does that then speak to how we come up with vaccines for global use or either for personalized use? So there's a lot of conversation in the research community about personalized medicine, personalized vaccine use. But maybe one thing we can also start thinking about is making that one vaccine that might be great for everyone. And there are some examples, very old vaccines, not the recent one, but I think we have to begin to think more globally as we come up with new therapies or new vaccines.
And so that really bridges, I guess, segues into. So I just have one more question, and then we'll just check the group chat for questions from the audience. But really, how do we sort of bridge the the gap between the basic research that we do and then practical implementation, which kind of streamlines in from the previous question. And sort of the simple things like eating better, sleeping more, having like stress management. But I'm interested in, perhaps at the policy level, what are some things that we as scientists can help governments and sort of like, nations in revising or improving the approach to talking or even communicating with the public about ways to improve vaccine efficacy, especially with, I forgot the statistics, but there's going to be a lot of old people. It's going to be a lot of old folks within the next that 10 to 20 years. And so with that sort of not only economic burden, but also with the sort of like, it's going to be an incredible health burden, cost of care burden on hospitals and stuff like that. So how can we, what are some things that we can do as scientists in helping, I guess, easing the burden of aging?
I'll probably start with this, right? So the first thing I'll probably say, and not everyone might see it from this viewpoint, is to make a whole lot of noise, right? So a scientist, you're probably very well trained to generate empirical evidence within labs, but it really has very limited impact if people who are on the policy side of things, one, don't understand what it means, two, don't know where to put the resources that they have. So the first thing is really communicating these findings, and perhaps more scientists should be involved in policy making. I mean, being involved, speak to people in government. In recent time, we've had a whole lot of interest in terms of science communication, because we found that not only do people who are at the helm of making these policies not really understand what it sort of meant, people also in science are probably not doing enough to make them sort of aware of it. So and of course, this has downstream consequences. It means people at policy level need to put more money into clinical trials that are not just centered sort of on one demographics. And SARS-CoV-2 would be a classic example. A lot of the emergency use authorization it really did focus, if you look at the demographics of the populations, it really did focus on a very, very homogeneous population. Is 95% of sort of people were either Caucasians or people from that particular area, and the vaccines got emergency use for everyone around the world. Again, as we clearly indicated here earlier, you have genetic differences, you have age differences, you have population differences. So long story short, one key way to kind of probably make the first move would be scientists to get involved in more policy making, be there, speak to people in the communities, but also educate people at population level, because then they can also speak to people whom they've elected, i.e. their elected representatives. So that's probably a very low-hanging fruit that most scientists do these days. On Twitter, on LinkedIn, people talk about it. People make really nice infographics. People go on podcasts now, and I think this is just, people now understand that it's really important, because you have misinformation and you have disinformation happening, especially very well highlighted by COVID. So I think this is probably one of the low-hanging fruits in terms of how to just bridge that gap that exists at the moment.
Yeah, I think I'm in support of what Adam Riley said, and only thing, a few points that I'll be adding to it is that now we are into science communication, and the way we communicate the science, we should communicate it in an immature way. The reason why I said an immature way is that communicate the science using non-scientific terms, so that those other people in the other end will be able to understand. Most of these policymakers we are talking about, they are not scientists. So if we communicate in a scientific manner, they find it difficult to comprehend what we are really saying. And we should try to make our communication multilingual. So that most of the local diets we have in a different population, we're able to take the message to them. And I have learned a lot when communicating science, the language, choice of language on radio is different from the choice of language on TV, it's different from the choice of language on print media. So, and that's why we're having short courses now for scientists to learn how to communicate science to the appropriate population using the appropriate and choice of words so that the intention we have in mind will be rightly communicated.
I do also, support what my colleagues are saying, but for me, the key point here as a scientist is that we have now to train our student, or even to have some workshop in science to put more translational aspect in our project. So, which means, of course, you need to get funded, right? This is clear, which means this is what Adam was saying, that more scientists need to be in policy so that we have more funding and so on. But if you are not trained, then to put more translational aspect in your project, when you are drafting your project, so you cannot even think about communicating then after to the target population. So what we usually do, for instance, we have ongoing project, but then we always put in a translational aspect where we can go to the field to meet the population, and then at least connecting our lab data to how we can apply in the field. And then this is what we are doing. We are doing some treatment in Tanzania right now based on biomarker that we have identified in the lab and so on. So which means, for me, the key point today is to train scientists now to always think about translational aspect when they are drafting their project, which means even the funding body can even request from the project that you need to have a section on translation. It can be a communication to population, it can be whatever treatment, it can be whatever, but then should be inserted, implemented in that project a small translational aspect of your data. When you think about what can happen then after, why am I doing this thing? How can population benefit from your work, from your readout, from your data? This is a key point for me before you think even about communicating to the population or and so on and so on.
Yeah, I'll just add on really quickly. I think one of the ways is through what we're doing right now, right? We're communicating science, we're sharing, we're having conversation. I think the Immuno podcast that is hosting this with Black in Immuno is also doing a great job of doing podcast and encouraging science communication. So I think science communication is definitely key. And just to add on to what, well, I guess I have dissenting views about insisting that scientists have to include translational aspect to it. Like, I think science is very diverse. There are people that do basic science and it's totally curiosity driven, and that is important science as well. That is critical as well. And I think the advantage of being in science is you have freedom of exploring your ideas. The translation is fantastic, but we should have freedom in exploring ideas. But there is importance in translation, right? There's importance in doing translational research. For me, I think from a policy perspective, we have to engage more with the communities. We have to engage more with politicians. We have to make sure that there's a lot of division in the world, but that we see ourselves as a global village, that everybody is important regardless of where they come from. And so we're including that perspective in science, that it's a global world, and everybody, if you have a particular pathogen or infectious agent, is affected regardless of where they are, and putting that in perspective in policy making, especially from the politician perspective. That is, we're not in an isolated world, right? It's a global village, and so we have to communicate and help one another in fighting this pathogen, infectious agent. I think that's something else I would add into.
Yeah, I'll totally agree and second that, because science is heterogeneous, and we have colleagues where their interests are only basic sciences, and they spend many, many years there. And really, sometimes that kind of forms the basis, whether if, for example, you're working in the HIV field and someone spends 10 years developing dolutegravir, for example, and then you can take this and at population level, give it to everyone, and you can measure suppression rates after 12, 24 months. So every project probably will not necessarily have, you know, short-term translational outcomes that can be measured using simplified metrics, I think. And I think it's just important to probably just to highlight that in that sense.
I mean, I think it's funny. It's not necessarily funny, but in my head, the things that unite us, aliens, alien attacks, and pandemics. Pandemics definitely forced large majority of the world to work together. The COVID-19 is the most recent example. This discussion has been amazing, and I'm hoping the folks in the audience and the folks who watch this recording later, you know, get some of the some of the understanding that we're trying to provide. And so I'll just instead of me asking more questions, I'll look at the group chat and see who's asked anything. And so there's one question so far from Nathan Ohio Makari, and this is a very basic question. Basic is in the sense of science. So he says, in a study design, if I were measuring efficacy of HPV vaccines in HIV positive girls, doses 0, 2, 6, and 12 months. One, what would be the best way to measure efficacy? And two, what would be the best regimen, dose spacing for an efficacy measurement? It sounds like a grad school question to me, but who wants to, do you guys have any perspective on this one?
Well, I don't know if there's any HPV sort of experts in the field, but I've done a couple of clinical trials in that sense, not necessarily as it relates to HPV, but so certainly as it relates to sort of Hep B vaccines and whatnot. And under these circumstances, it will really depend on essentially what you're trying to measure. For example, if you're giving someone an HPV vaccine who's HIV positive, you probably want to look at the number of people who are protected, i.e. they don't pick up an incident HPV vaccine. And again, it would really depend under these measures, and it would absolutely depend on what your intended outcome is, i.e. primary and secondary outcome. So there's probably no direct answer for you in this sense.
Anybody else?
So I'm not really in HIV and HPV, but I mean, so globally or in principle, you want to measure some readout related to HPV vaccine, right? Which mean, I don't know, whether you have specific antibody and also T cells and so on in non-HIV, and then in HIV girls. I mean, it's girl, right? Exactly, in HIV girls and so on. There have been, maybe I can recommend to Nathan. So there is a paper published by Ellison Elliott from UK, and they really run a lot of study in Uganda, Cameroon, and also somewhere else where they have they test then I cannot remind if it is HPV, but then TB vaccine and so on yellow fever vaccine and so on HIV infected, helmet infected, and so many in different settings in rural and then also urban setting where they analyze the efficacy of the vaccine in comparing those two setting or environment. And then I do remember that they use antibody, they use effector T cells, they use also even regulatory T cells. As I mentioned, it depends on your intended objective and analysis. They even use regulatory T cells, which supposed to not be a readout in case of vaccine efficacy. But then since they have schistosoma or manzoni, and so which is known to induce a lot of regulatory T cells, they also put this readout. So maybe just type Ellison Elliott and vaccine, yellow fever vaccine, TB vaccine, and so on. And then you're gonna find really they have a really nice study design and clinical study design, how they move from one step to another one, and so on and so on. So this is what I can say and also suggest here. I'm not really in that field, so I cannot say much about that.
I mean, I think that's more, again, because we're not experts in everything, but the fact that, you're able to provide both you guys are able to provide some references and some perspective is good enough. And again, the audience, if you, the reason for these sort of forums, thanks Black and Immuno and African Immuno podcast as well, for hosting this sort of like discourse, is because we want to be more accessible to the public. And so with that said, are there any final thoughts from the panelists that you want to share?
Who starts? Should I start? Or?
I guess I can just say that whoever is listening to this is an opportunity for you to also talk to your parent, your grandparent, and share the idea that vaccines do work. And of course, there is variation in the population. There are people that do get vaccine and get protected, and there are people that do not for some reasons that we've discussed today. So age is one of them, environmental factors, what you've been exposed to in your lifetime. So there are variation in vaccine responses. And this, just to kind of coming back to the idea that you have different miscommunication and dissenting voices out there. So like if you're listening to this, share the word vaccines work. There are variations, but scientists are looking into that, and we're encouraging more people to get into research, into the STEM field, to kind of help us as a global community to answer some of this question. I think that will be my last word.
Maybe I can add something on that. I mean, today's section was about immune response, immunity, aging, and then also infection and so on. And so as my colleague mentioned, of course, we should now, because so older people, they are explicitly really excluded from clinical trial. So many of them, they are quite excluded from clinical trial for many reasons, to be honest. But then nowadays, what we are seeing is, so when you have aging, and then we are developing so many measures then to have a longer life, but then this is not parallel with health span, right? So it's quite different. And then the better way is now to include really older people more and more into clinical trial, and especially to really reflect on how to target cell exhaustion in older people, because the main problem in elderly population is the cell exhaustion, because the cells are not really fit to react, or they are too tired, they are too old. Is now to reverse this. I mean, so Adam already mentioned PD-1 as a checkpoint. This is now recognized today as a cell exhaustion marker in CD8 T cells and so on. So, which means we need to really involve more and more the older people in clinical trial, and even funding really targeting the health of the older people, to design better vaccine for them, to fit their immune system into a way that can react better to vaccine and protect them from infection and pathogen.
Yeah, so some final words for me, perhaps, that we've really talked about a whole lot about vaccines today, and perhaps one of the greatest discoveries in modern history would probably be vaccines. It's really allowed for humans to live significantly longer. It's certainly averted a whole lot of morbidity and sort of mortality. And I think that the key point here is that vaccines do work. There's a lot of misinformation and disinformation out there, but there's also a whole lot of sort of science communication platforms that go on to talk a whole lot about this in simplified terms. So please do listen to them and talk to your family and friends about them. And also if there's anything, I'm pretty sure probably for the rest of the people here, we're relatively, you know, available. So you can certainly reach out via email if you have sort of any questions, if you have any interests. And I think generally speaking as well, speak to people both in the scientific world, but also people in the general audience are typically the bridge between what scientists do and what people on the other side can also do. So please do use social media platform to reach out and communicate to the rest of the world, I think. Thank you very much.
Yeah, I will start from where my brother ends and to let sure that vaccine works. And one of the wrong perception that I have tried several times to correct that is that because you are vaccinated doesn't mean you cannot be infected. So that is one bad ideology that people had in mind that, oh, because I'm vaccinated, so no infection at all. So, and that's what the problem we had during the era of COVID, while some were talking about things that are not right. And we should always remember that we also have a group of people called anti-vax, that they do not support vaccine at all, despite there are a lot of data sets generated showing the efficacy and how effective vaccine is. So we should go with data-driven science that is driven by data, and that is a correct peer-reviewed data that is supportive what is in existence. And in Africa per se, among the Black population, after vaccine has been given, I want to encourage the pharmaceutical industry not to neglect the post-vaccination surveillance, that it should be ongoing. The work has just started. And that's why Lee was talking about the clinical trial. A lot of people have been vaccinated, either for one pathogen or the other. After certain years, how are we measuring what is going on? Or go to the population, approved, given, and the next 3 or 4 years, nobody's checking what is going on among the vaccinated population. So we need to do our continuous research, like Dr. Oyebola rightly said. That is part of the basic research to contribute to the knowledge pool, that after a certain number of years you have gotten this vaccine, what is the response look like? What are the other things to do? What are the things we need to improve? Science keeps improving, and I believe that by the next time we are having this kind of discourse, there will be a greater improvement to talk about. Thank you again.
And so I'll do one final thanks to all the panelists. Thank you for taking the time. And I mean, I really enjoyed this discussion. Final thought is that vaccines work, like they really do. So take your vaccines if you take your vaccines. With that said, I know there's some folks posted some questions after the fact in the group chat, but don't worry, we're gonna answer those on Twitter. But for the sake of time, I want to thank all the panelists. I want to thank everyone for their attendance. And yeah, we'll catch you next time for the next podcast. Let me know. Okay. All right. Well, you guys have a good day.
Thank you, Lord.
Thank you for having us.
Thank you for having us.